Hi, this is Dr. Bosch, and I want to take some time to go through the steps of muscle contraction with you. One of your essays on your next exam will be asking you about these steps, so it's very important that you understand this kind of complicated process. So we'll take it one step at a time. This slide shows you the actual steps of muscle contraction, but as you can see, it doesn't really start the process until a signal comes from this object, which is a neuron, the axon of a neuron. So I won't take a long time to explain the role of the neuron, but we do have to have enough information that you can understand how this whole process starts. So I'm going to back up a little and say, how did we get to the point where a neuron was stimulating a muscle cell to contract? So here's a neuron. You saw those when you were looking at the tissue slides. And a typical neuron has a fairly large cell body here. There's the nucleus. And then dendrites, which generally take information in. So information comes from the end of these dendrites into the cell body. And then that same message continues along the axon. So if we look at impulse direction, it's moving in this direction. Down on this end is the end of the cell. And for our discussion today, this is where a muscle cell would be. A muscle cell would be attached to these terminal branches right here, what we call a synapse. Now, we're going to focus on this part of the neuron, the axon, because that's where the signal moves from this area of the cell out to the muscle. If we look at that same axon at rest, we note that there are various ions both in the axon, that's the inside, and outside the axon. We'll be going through this in detail when we do the nervous system, but for now, you should know that the outside of the axon is more positive, the inside is more negative. We say that this resting axon, so it's not doing anything, it's just resting, has a charge of minus 70 millivolts, which means that it's polarized. So a resting axon like this is polarized. Something else to note for now, again, not looking at too much detail, is there's a lot more sodium on the outside that's being prevented from entering because there's no pathways. About 150 millimoles, here's only 15. So there's 10 times more sodium on the outside than the inside. And the opposite's the case for potassium. 150 millimoles here, only 5. That's 30 times more potassium inside the cell than outside the cells. This is the setup of a resting membrane when it's not being used. When we use that neuron, in particular what we call a motor neuron, one that's attached to a muscle cell on this end over here, the signal will pass from the dendrites to the cell body and then down the axon. This is a close-up of the axon. When the signal reaches this little place right here, the signal causes the gates on the outside of this axon to open and sodium rushes in. So all those positive sodiums rush in, changing the charge. If you look now, it's positive on the inside and negative on the outside. That's the opposite of what it was when it was resting. So we say there's been a depolarization. Depolarization is like an impulse where the sodium rushes in and changes the charge. Now, when the sodium rushes in like this and makes the inside of the cell more positive and the outside more negative, that allows potassium to leave. It's also kind of stuck in here because of the environment inside and out. Pluses don't like pluses, they repel. But now, with a negative charge on the outside, the potassium can rush out. And it'll keep rushing out until it's back to normal. It's back to its resting state. So the basic process here is that when an impulse reaches this part of the axon, sodium rushes in, potassium rushes out. Then right next to it, sodium in, potassium out. Sodium in, potassium out. And this goes all the way down the axon, so all the way down here to the very end. Sodium in, potassium out, sodium in, potassium out. And that is a nerve impulse, those changes in charge. So what I'd like to show you next is what happens when it gets to the end, down here, these axon terminals at the very end. What happens is that charge, when it gets down to the very tip here, what we call the axon terminal, that will cause calcium to enter this terminal. So the change in charge, right, the positive sodium's rushing in, that change in charge causes calcium to come in, and then this axon terminal releases a neurotransmitter called ACH. ACH. And ACH will attach to the muscle. This part is muscle, the pink is muscle, the yellow is a neuron. It causes this muscle to also depolarize. In other words, the muscle cell is a lot like the neuron in that it is polarized at rest, but the ACH that comes from the axon attaches over here. Here's a close of it. It's attaching on this side, allows sodium to rush in, causing a depolarization. Now, if you don't like to say depolarization, you can call it an impulse. It's a muscle impulse. So this tissue right here gets depolarized. What causes it? The neuron. That's why we have to have a functional nervous system in order to make our muscles work. In fact, if anything happens to our nervous system, such as breaking our spinal cord or something like that, our muscles no longer work. We need that. So this neuron is causing this muscle to contract. Our next slide is when the rubber hits the road. Our next slide actually shows the contraction. Oops. Here's that slide. It had kind of disappeared on me for a minute. So here is the neuron, here is the muscle. What happens to allow the muscle to contract is the ACH comes across the ACH. That causes sodium to go into the muscle cell, depolarizing it. And just like in the neuron where it's depolarized here, then depolarized here, then depolarized here, right down the axon, same thing happens on the muscle. Now this muscle cell has a sarcolemma on the outside. That's just a cell membrane, but a cell membrane of a muscle we call a sarcolemma. And so the sodium's rushing in, the potassium out, keeps going like this, moves across the outside of the muscle cell, and then goes down into what's called a T-tubule. It's a transverse tubule that brings this signal, brings this depolarization deep inside of the muscle cell. So this is all muscle cell right here. As this impulse rushes past this endoplasmic reticulum, you may remember that from earlier biology classes. We actually call it sarcoplasmic reticulum in a muscle, but it's basically a membranous body that happens to have calcium inside. As this signal passes by the sarcoplasmic reticulum, it causes calcium to be released. So there's all these calcium ions being released by the SR, by the sarcoplasmic reticulum. That calcium will go over to the molecules that are on the actin and myosin. A quick reminder, the fibers found inside muscle cells are called actin, those are the thin ones, and myosin, those are the thick ones, and they're right next to each other. You can check that out another time. Anyway, the calcium will bind to troponin. That's a kind of a molecule, protein molecule on the outside. And troponin changes the shape of tropomyosin so that this part of the myosin, this cross bridge, can attach. So without this signal, without the neuron doing this, and without this signal, and without the calcium, the actin and myosin just sit there doing nothing. They can't interact. But in the presence of calcium, this myosin head can attach to actin and move it. That is a muscle contraction. The cell is actually becoming smaller. So, for example, when you flex your arm, when you pull your hand close to your body, the reason it's getting closer and closer is your muscles are getting shorter and shorter. They're contracting. So, the contraction occurs, and then it takes energy to move that calcium away and put it back into the sarcoplasmic reticulum to be used later. Now, once the calcium is removed, the contraction is over. And what happens is the tropomyosin will block the myosin from the actin one more time, so the muscle's not being used, and then the calcium is put back into the sarcoplasmic reticulum. So let's go through those steps one more time. You do have to have a nerve impulse to make this whole thing work. So keep that in mind. We need nerves to make our muscles work. This is showing just one cell of the nervous system, one neuron. And the signal moves down this axon, this part of the neuron called the axon, to the end. So sodium is rushing in, potassium is rushing out. The impulse gets to the axon terminal, which is the end. That causes calcium to go into this terminal. Calcium causes ACH to get spit out. It's a neurotransmitter that comes across to the muscle side. That's neuron, this is muscle. Opens up this membrane so sodium can rush in here. And of course, potassium back out. So that is the impulse. So it's an impulse that comes all the way down the neuron, gets transferred at the synapse to the muscle, and then goes down the outside layer of this muscle cell. We call it a muscle fiber because they're long and stringy like a fiber. Now, that action potential, that impulse, will go deep into the muscle. We're only showing one little part here to make it easier to see, but this is happening all over on all sides of the muscle. The signal is going deep in the muscle along a T-tubule. That impulse causes the calcium in the sarcoplasmic reticulum to be released, and the calcium will come over to the actin and change its orientation so that the myosin and actin can interact, so that a contraction can take place. The details of that, and you do need to know those details, is that the calcium binds to a molecule called troponin. Troponin changes shape. That removes the blocking action of tropomyosin. In other words, we don't want our muscles to contract all the time. You don't want to swing your hand if you're just sitting here. You don't want to move your neck. But when you do want that muscle action, this whole process has to occur, where the troponin changes shape, moves the tropomyosin out of the way. This myosin head pulls the actin that way, and there's a muscle contraction. When it's over, and it's going to be a matter of less than a second, when it's over, the calcium is removed from the troponin, the blockage comes back, and the calcium is placed back in the sarcoplasmic reticulum. So I hope this was helpful. I would definitely view your textbook, read this in detail, and maybe practice writing this out as far as the steps go, because that's what you have to do for your essay. I hope this was helpful, and I will include these drawings.